I'm very pleased to introduce Professor Luisa Lanfranco, full professor at the Department of Life Sciences and Systems Biology at the University of Turin, where she also coordinates a PhD program in biological sciences and applied biotechnologies. She earned her PhD in biology and biotechnology of fungi from the University of Turin in 1998, working under the supervision of Professor Paola Bonfante. She began her academic career as an assistant professor in 1999 and became an associate professor in 2005, finally also a full professor in 2023. Over the past almost 30 years, Luisa has focused her research on plant-fungus interactions, especially arbuscular mycorrhizal symbiosis, a fundamental and ancient mutualistic relationship. Her work has explored the biology and genetic complexity of these fungi, including their microbiota and the molecular mechanisms that regulate their interaction with plants. She has authored over 100 scientific publications, contributed to national and international projects, and received recognition during the Assunta Baccarini Alumni Award. She is also a passionate science communicator and has coordinated the Fascination of Plants Day in Italy since 2018. Luisa is also a member of several prestigious scientific bodies, including the Italian Society of Botany as a board member and the International Mycorrhizal Society, where she served as a secretary of the advisory board. She also contributed to the Italian Space Agency's roadmap of biological life support, so all very interesting stuff. So without further ado, Luisa, the floor is yours.
Thank you very much, Naomi, for this very generous introduction. So, hello to everybody. And today I would, and I thank you for the organizer, the Root Benefit cross-section, for giving me the opportunity to present our group and what some of the research we are doing here in Turin. So we are placed in Turin, so the university, so northwest Italy. So the university is rather ancient. So now we have several first and second level courses and more than 30 PhD programs covering almost all the research areas. And here you see the symbol of our cities, which is the Mole Antonelliana, just from a view from the River Po, that also cross the city. And our laboratory are within this historical building, very close to this downtown, these other places. And this building is inside the botanical garden. And I can say that within this historical building, a little bit of history of mycorrhizal research was made. So long ago, in 1883, so Giuseppe Gibelli, who was a professor in botany in our university, provided this beautiful drawing of chestnut roots showing the typical feature of ectomycorrhizals. And a few years later, always in Turin, Oreste Mattirolo hypothesized for the first time that truffles, so the gourmet specialty also typical of our region, were actually formed by fungi living in symbiosis with the trees. And he committed this painting showing a man together with a pig and a dog looking for truffles under an oak. And you can see here the roots of this oak is display again the typical features of ectomycorrhizals. But moving to more recent times, not only ectomycorrhizals, I have, of course, to mention Paola Bonfante. Maybe many of you know her. She is now an emerita professor at the university, and actually she dedicated all her life in the study of plant microbe interaction, in particular mycorrhizal interaction. And she first gave description of endomycorrhizals. In particular, she started her research career in the golden age of electron microscopy, and when looking at the features of the fungal cell wall across the different stages of the fungal life cycle, moving from spore to extraradical hyphae to intraradical hyphae, she could see that even the finest fungal hyphae inside the plant cells are always surrounded by a plant plasma membrane. So this knowledge is still valid today and is a hallmark, a defined hallmark of mycorrhizal interactions. So since then, Paola did many things and many studies. She used different cellular and molecular biology approaches to study mycorrhizal interaction and cover and consider all the different components. So not only plants and fungi, but also bacteria, especially those living inside arbuscular mycorrhizal fungi. And so you can see here Paola at the very early in her career, in the first meeting devoted to endomycorrhizal, which was held in Leeds in 1972. And you could see from this photo also many other top scientists in mycorrhizal research. So I have to say that the group we have now in Turin was actually deeply influenced by the activity of Paola's research. And indeed, she, as Naomi was saying, she was the supervisor of my PhD thesis, actually my mentor. But this was also the case for other people in our lab. So now the group is rather large. Here you see the people that are currently in staff. And we are all working on plant-microbe interaction, in particular endomycorrhizal symbiosis, considering arbuscular, ericod, and orchid mycorrhizals. And we are developing more basic research, I would say, looking at the cellular molecular mechanism of plant colonization and the plant and fungal responses to the interaction, but also in more general, we are also looking at the taxonomic and functional biodiversity of the plant microbiota. Some of us also developed some more applied research with the in-field application of microbial inocula and the screening of, let's say, microbial plant growth promoting traits. So my current research line mainly focuses on Arbuscular Mycorrhizal symbiosis. We are investigating the role of carotenoid-derived compounds, mainly apocarotenoids, and the impact on plant biology, and especially on mycorrhizal interactions. We are also studying the microbiota of AM fungi. In particular, in the recent time, we focus on the description of the virome, so the viral sequences present inside AM fungi. But today I would like to present some of the work we have been doing in the last year on the small RNA. And I did this work in strict collaboration with the colleague Valentina Fiorilli. So just a brief introduction for the AM colonization. So it involves different steps. During the pre-symbiotic stage, we know that the fungus and the plant release diffusible molecule that allow the reciprocal recognition. So once the fungus reaches the surface of the root, it develops a penetration structure, which is called a hypopodia, and then the fungus develops inside the root through inter as well as intracellular structures, and inside the inner layer, I would say, of the cortex, the fungus develops this very highly branched structure, which are called arbuscules. So arbuscules are the key sites of nutrient exchanges. As I mentioned before, in this situation, the arbuscules, the hyphae, even the finest fungal hyphae, are always surrounded by the so-called periarbuscular membrane, which is derived from the plasma membrane, the plant plasma membrane. And on this plasma membrane, we can find the transporter which is involved in the uptake of mineral nutrients such as phosphate, nitrate, ammonium, that are efficiently taken up by the fungus from the soil solution and then delivered to the host plant through the so-called mycorrhizal pathway. Also on this PAM, we can also find transporters which are involved in the delivery of carbon compounds from the plant to the fungus, sugar and mainly fatty acid. So several signaling molecules have been described as involved in these interkingdom interactions. In the root exudae, strigolactone are the best, say, the most studied signaling molecule that are able to activate the metabolism of AM fungi, which in turn release the chitin-based molecules, which are also considered important to start to elicit the symbiotic plant responses. But this is not the only signaling pathway. We know also from the seminal studies of Caroline Gutjahr that there is another signaling pathway which is based on the alpha-beta fold hydrolase called D14-like, which is essential for the establishment of the AM symbiosis in rice, but we still don't know which is the ligand, which can be of plant or also fungal origin. So we also know that in the genomes of AM fungi, we have many genes encoding for putative protein effectors, and only a few of them have been characterized so far, and some of them have been shown to have a role in symbiosis formation. We also know that AM fungi release phytohormones, as you can see here from these pictures, but we still don't know which is the role, if these hormones can play a role in the interaction with the host. But some years ago, we hypothesized that in this symbiotic interaction, also RNA effectors could play a role, based on the studies, on the results that were made in plant pathogenic systems. And so, in particular, we hypothesized whether small RNA could be involved. So small RNA are short RNA molecules that act normally as a negative regulator of gene expression. And by exploiting a complex process, which is called RNA interference, RNAi, and how they act, this small RNA, they can target in a sequence-specific manner genomic DNA, leading to the formation of silent chromatin. And so this is a transcriptional silencing, or they can target RNA molecules, inducing the cleavage of the messenger RNA or the inhibition of its translation, leading to a transcriptional or a post-transcriptional gene silencing. So this RNAi process is dependent of the activity of three main classes of protein, the dicer-like, which are involved in the production of small RNAs, starting from long double-strand RNA or hairpin RNAs. Then we have the key components of the argonaut. The argo proteins are able to bind the small RNA, and through this binding, they can target and recognize specific sequences, DNA or RNA, and then they start the induction of the silencing. And then there are also RNA-dependent RNA polymerase, which are involved in the amplification of the silencing signal. So small RNA and the RNA interference were first described as working with an endogenous role, working in the same organisms that are producing the small RNA. But in more recent times, small RNA were also shown to play a role in inter-kingdom interaction. Some small RNA, in fact, can move, are mobile, and can move from donor to receiver organisms, where they can regulate the gene expression of the receiver organism, exploiting the RNA interference machinery in a process that is called cross-kingdom RNAi, when, of course, organisms belonging to different kingdoms are involved. So first studies were published on the plant pathogen interactions, but more recently, example of cross-kingdom RNAi were also obtained from mutualistic interactions. For example, it has been shown that small RNA from Rhizobium are important signaling molecule controlling plant modulation. Also, the ectomycorrhizal fungus Schizolitus was shown to produce and deliver a microRNA to the host plants, and in this way control the symbiosis with the host plant. And also, I would like to also underline that in more recent times, some data on the possible cross-kingdom RNAi were also obtained from other, let's say, endophytic, fungal endophytic interactions. So, but what about the arbuscular mycorrhizal symbiosis, which is one of the most ancient and the most widespread symbiosis, root symbiosis, fungal plant symbiosis? And so we have some indirect evidence. So we know that extracellular vesicles are formed at the periarbuscular space, where the fungus and the plants live in a very intimate association. And as far as we know from at least some of the plant pathogenic system, we know that small mobile RNA can use extracellular vesicles to move from one organism to another one. Then, in addition, we know that host-induced and virus-induced gene silencing have been used as a successful tool to silence fungal gene expression in mycorrhizal roots. So, pointing to the transfer of small RNA from the plants to the fungus. And also, more recently, a nice work from Xie and Xie demonstrate that the AM fungi can also uptake double-strand RNA from the environment and use this double-strand RNA to modulate its gene expression. So at the time some years ago, we were interested to see whether small RNA could act as signaling molecules in the communication in the AM symbiosis between the fungi and plants. And we gave attention to the possible transfer of small RNA from the fungus to the plants. But first of all, we had to prove whether AM fungi could produce functional small RNA, whether the AM fungi possessed all the components for the RNAi machinery. So we started this characterization by looking at the model AM fungus Rhizophagus irregularis. And by looking for proteins with the conserved domain, we could see that Rhizophagus genome contained all the typical components of the RNAi machinery, but with an unusual expansion of EGO-like and RDRP gene family, as you can see from this table. And by analyzing in RNA-seq data sets and also doing some RT-PCR experiment, we could prove that the majority of these genes identified in silico are actually expressed, although so far none of them have been characterized in details. Then we analyzed by next generation sequences the population of small RNA in the specific association between the host plant Medicago truncatula and Rhizophagus irregularis. We characterized the small RNA from the exoradical mycelium, mycorrhizal roots from which the exoradical mycelium was removed, and this sample corresponds to the intraradical phase, considering the fungal site. And then we also analyzed control non-mycorrhizal roots. And by looking at the small RNA mapping on the fungus, we could identify more than 2,000 small RNA generating loci. And based on the specific features, considering the length, considering the genomic position, the origin of the genomic position, considering the expression profile, we could create two different clusters. So the first one include loci producing small RNA of different size, mainly overlapping with protein encoding genes, but not differentially expressed between the exoradical and the intraradical mycelium. And the second cluster containing loci producing small RNA enriched in the 22-24 nucleotide, and then corresponding to intergenic region. And interesting, many of these small RNA were also upregulated in the intraradical phase. So based on the features of this small RNA and considering the identification of the RNAi related genes in this fungus, we could say that indeed the Rhizophagus, this end fungus, is able to produce functional small RNA. So we wanted then to see whether some fungal small RNA could potentially target transcript within the transcriptome of the host plant Medicago. And so first we selected the small RNA generating loci upregulated in intraradical mycelium. Then we identified the most abundant small RNA in each locus, because we had many for each locus. And then we used this in silico approach to make an in silico prediction of putative targets in the host plant. And we found a number of fungal small RNA that could have potentially targets in the plant transcriptome. We focus in particular on this one, 2216, which is 21 nucleotide long, because it was upregulated in the intraradical mycelium compared to the exoradical mycelium. It had a high number of potential targets, notwithstanding despite the high stringent parameter that we set up with this in silico bioinformatic analysis. And then because it was also identified in other RNA-seq data sets from Rhizophagus irregularis colonized roots. And among let's say, the best hit of this in silico prediction, we found these genes encoding for a transcription factor belonging to the WG2 family. This was named WG69 because there are many, many different members of this transcription factor family. And we focus on this one because it is known that many transcription factors belonging to this WG2 family are involved in the responses to biotic and abiotic stress. And so, but for this was only an in silico prediction, so we had to generate experimental data confirming the potential interaction between the fungal small RNA and the putative target WG69. So at first we set up a co-expression assay in Nicotiana bentamiana, and when we expressed the putative target WG69 in fusion with the YFP, we could see a strong signal corresponding to the accumulation of the protein in Nicotiana bentamiana leaves in this northern blot using antibody against the YFP protein. But when we co-expressed together with this gene, the fungal small RNA 2216, we can see the completely abolished of the accumulation of the protein. And this was not occurring when we co-expressed another small RNA, which was not actually recognizing any sequence in the WG69. So with these results, we could say that 2216 is in principle to be able to silence its predicted targets in WG69 in plant in a sequence-specific manner. But then again, this system is a little bit artificial because we are overexpressing with a constitutive promoter both the putative target and as well the small RNA. So we move to our biological system, so mycorrhizal roots. And at first we look at the expression profile of this WG2, and we took advantage of this technique, the laser micro dissection technique, to collect arbuscule containing cells from roots, from mycorrhizal roots. And in parallel, we collected cortical cells from non-mycorrhizal roots. And to verify the quality of these samples, we analyzed the expression profile of the phosphotransporter 4, which is considered a marker of arbusculated cells. And you can see an increase in arbusculated cells. And when we look at our WG69, we could see a downregulation of this gene in arbuscule-containing cells compared to cortical cells from non-mycorrhizal roots. Then we also used a 5' race assay and in mycorrhizal roots of Medicago truncatula, and we were able to detect the truncated fragment of WG69, which was exactly cleaved within the predicted binding site of 2216. So this is an important evidence that this putative target is cleaved in the specific site recognized by the fungal small RNA. And in addition, then the question was, but is 2216 able to move to the plant cells and to maybe associate with components of the RNAi machinery in mycorrhizal roots? Of course, we set up this co-immunoprecipitation assay by using an antibody against the AGO protein that allows us to immunoprecipitate not only the AGO protein, but also the small RNA associated to this protein. But at first, we have to decide which AGO1 consider for this experiment, because in the Medicago truncatula genome, there are at least 12 different AGO genes. So we consider the AGO1 because it preferentially binds 21 nucleotide long sequences that present a uracil at the 5' end. And these are the feature of our 2216 small RNA. And then these homologs of these sequences were also found to be involved in cross-kingdom RNAi, in plant pathogenic interaction, and also in the Rhizophagus symbiosis, and also because AGO1 turned out to be upregulated in our arbusculated cells. And so, but first we had to, let's say, prove our ability to detect the small RNA 2216. And at first we set up this stem loop RT-PCR experiment, and we tested this approach on RNA from mycorrhizal and non-mycorrhizal roots. So you can see we could detect this small RNA together also with other small RNA belonging to the fungus, and also we could detect also as a positive control of the non-mycorrhizal sample small RNA from the plant side. But interestingly, we could detect this 2216, the fungal small RNA, also in immunoprecipitated fractions, which was obtained by using an antibody against the AGO1. But in this immunoprecipitated fraction, we could not detect two other fungal small RNA, despite they were highly abundant in the, let's say, whole RNA from mycorrhizal roots. So this experiment clearly showed that 2216 can associate rather specifically with the host AGO1 in mycorrhizal roots, pointing to the possibility that it moves from the fungus to the plant and exploiting this AGO1 RNA machinery to downregulate the WG69. But of course, we had to also have evidence of what was the role of WG69 in the M symbiosis. And so we generate plant overexpressing the WG69 in parallel to plants used as control overexpressing the fluorescence protein scarlet. And at first, we had to, of course, prove the overexpression of this sequence, and then we analyze the, let's say, mycorrhizal phenotype of this WG69 overexpressing plant. As you can see from here, this is a molecular analysis, always detecting the abundance of the marker, the phosphotransporter 4. As you can see, we have a strong lower level of ion colonization in the WG69 overexpressing line compared to the control line. And this molecular data was also confirmed by the morphological analysis of different parameters concerning the morphological analysis of frequency, intensity of arbuscular abundance in these mycorrhizal roots. And so this data is also showing that the expression level of WRK69 is an important factor to control the extent of the AM colonization. So to sum up, we were able to demonstrate that this AM fungus possesses an RNA silencing machinery, and it can produce two different populations of small RNA, which are likely functional small RNA. By an in silico prediction software, we could find that some of these fungal small RNA can potentially target transcript within the transcript of the host plant Medicago truncatula. And by focusing on the 2216, we demonstrate that this fungal small RNA is able to move to the plant, bind to the AGO1 component of the RNA silencing machinery of the host plant, and to downregulate the WRK69 proteins. And this is the first evidence of an event of cross-kingdom RNAi in the fungus to plant direction. And we are now actually working on to understand how this WRK69 is controlling the extent of root colonization. So with this, I would like to end and thanking the people directly involved in this research from my lab, and also very fruitful collaboration we have in foreign countries. And of course, I thank you for your attention and for being here in such a holiday period, and will be pleased to answer your questions.
Thank you, Luisa, for a very clear and very interesting presentation.
I hope that can
really and just there you go. Yeah, so again, feel free to ask any questions in the chat or to activate your microphone. I already have a question to get us started. Yeah, it's fascinating to see how little is known about this still and how much potential there lays. And what I'm wondering is it like somewhat known how these small RNAs are targeted to the vesicles, how they're loaded into the vesicles and such?
No, actually, it is a really still open debate, because some groups were actually showing that the small RNA are loaded inside these extracellular vesicles. This was shown by the group of Heilingen in the pathosystem between Arabidopsis and Botrytis genera. But other groups are actually showing that the situation is much more complex, because we can have small RNA inside, we can have small RNA attached on the surface of extracellular vesicles, but we can also have small RNA bound to RNA binding protein, and even double-strand RNA free outside the cell. So which is, it seems a rather weird situation, because we are always thinking about RNA as a very fragile molecule. But indeed, it seems that there are some very nice papers published by the Roger Eames lab this year, showing that there is really a much more very complexity in this delivery in where is the extracellular RNA. So it can be associated inside, associated to vesicles, but also outside, maybe only associated with the RNA binding protein or even naked RNA. So there is still a very open debate on this in the scientific community working on extracellular vesicles.
It's fascinating to see what research will come out on this. Then we also have quite a few questions already in the chat. So we have Benoit, for example, that asks if you have any explanation for the high number of argonauts in the Rhizophagus genome, and have you compared it to other mycorrhizal genomes and such?
Yeah, so in the first work we published in 2019, we also compared, because at that time the Rhizophagus clarus genome was also available, and we could see similar number of AGO genes. I have to say that we consider all the AGO-like. So because out of the 40 AGO-like protein we found in the Rhizophagus irregularis, only 25 had the 4 typical domains of the AGO proteins. The remaining have at least the PW, which is the key functional domain of AGO1, of AGO. And so this is not easy to answer because we cannot do genetic transformation of M. fungi, so it is very difficult to really characterize in details these in silico annotated genes. And yes, but also we analyzed the situation in Gigaspora margarita. We could find a little bit less, about 11 AGO-like protein, and out of 11, 5 had all the 4 typical AGO domain. And the other one lacked one of these typical domains. But also in this case, we actually we are missing a detailed characterization of them. We speculate why such a huge number of AGO-like genes. We know that the genomes of AM fungi are very rich in transposable elements compared to other fungi because they have very large genomes. We can have much larger than other fungi, the average of fungi. And so these probably may be having such a high variety of AGO protein, which can be involved maybe in the silencing of these plethora of transposable elements. But this is really just a speculation. Yeah, cannot say more.
Again, very interesting. And then also another related question of Benoit was if you can do a similar speculation about the number of small RNAs encoded in Rhizophagus irregularis and then compared to the other phyla as well. Do you have any idea of that?
No, we didn't do this comparison, but it is also the word of small RNA and RNAi in fungal kingdom is very peculiar. It's not like it happens in plants where you have maybe a number of small RNA which are very conserved across plant families and so on. So it seems that in fungi, also if you compare the very close, phylogenetically close fungi, they can have a completely different small RNA with completely different features. For example, just referring to the length. So there was in the description of similar genera, fungi belonging to the same genus, that shows small RNA with different length. So it's very, we still know very little on how this small RNA and RNAi is working inside the fungi, and it's even more complex in AM fungi. So yeah.
And then Juanan also asked a question, this time related to the worky that you found. Do you also have a knockout phenotype?
We have seeds of this knockout we received from the USA, but we have to grow them to generate the homozygotic line. So far we don't have any data on knockout, WK69 knockout lines, unfortunately. But we are trying to, identify the genes which are targets of WK69 because we know it is a transcription factors, and so it possibly regulate the extent of root colonization by modulating the expression of some target genes. And so we are doing some experiments trying to understand which genes WK69 is actually regulating during the mycorrhization.
Mm-hmm. And can you speculate on if you think that this may be defense related, for example?
Yes, this was our first idea because many WIRKI genes, although this WIRKI69 from Medicago has not been characterized, although there is from the atlas, in the expression atlas, we could see a regulation also during nodulation. So this was also pointing us the interest on these genes. But it has not never been characterized in details. So this is one of the possibility, yes, is that it can control some of genes related to defense or immunity, and maybe to balance. We know that, especially from the Benoit works, that we know that the plants should balance between immunity and symbiosis to be able to recognize the fungus and allow it to accommodate it inside the root tissue by limiting the access of pathogen, for example. So yeah, this could be a possibility.
Very fascinating. Then we also have a question by Yusuf, who is asking if you could provide any specifics on the in silico analysis also that you did for the prediction of the small RNAs, including modeling, simulation, and such.
Yes, I can. Oh, so this paper, the data I presented were published, the last one in New Phytologist. So if you want, I can give you the papers and also the reference to these pipelines. Of course, for the target prediction, we used a software which was developed for plants. So we focus on the prediction of the targets on the plant side. But of course, having the small RNA also from the fungus, we could also investigate the possible intra-organism regulation by the small RNA. But as I mentioned you before, there is not really a consensus on how the small RNA are working in the fungal kingdom. So there is not a specific software able to recognize with fidelity, I would say, the targets, the fungal targets of fungal small RNA. So, and we used a software that was developed for plants. And actually we selected also small RNA which presented features similar to small RNA which are loaded on AGO1. So because we had a different one, but we could select the 2216 as is a 21 nucleotide long, which is and it has the uracil at the 5' end. So these are the features that are that really imply, let's say, a kind of affinity for AGO1, because the different AGO protein implants can have different affinity for different small RNA. So this is something that, yeah, why we focus also on this one.
Makes sense. Then I think there's still room for one final question since we're going a bit.
May I ask a question?
Yes, Sigmund.
Hi, Luisa. Yeah, hi, and
Hi.
Thanks very much for the elegant work. I really appreciated you sharing with us.
Thank you.
I was curious to ask, when you compare the extra radical mycelia versus the intra-radical, and you found when the candidate you showed us today, did you also look for other size small RNA species that could be like long non-coding RNAs up to, say, 150 or some transport RNAs that were differentially expressed?
Now, in that data set we generated already long ago, we focus on small RNA. So we do not have long non-coding RNA in that data set. So, but I agree that it will be very, very nice to very interesting to also to look for long non-coding RNA, because I think this is a kind of the dark matter of the old eukaryotic genomes. And so I think that also in these fungi and the interaction, they could play a role, definitely. But we don't have this long, let's say, longer than 30, let's say, we stop with 30 nucleotide long small RNA.
Thank you very much.
You're welcome.
Okay, so I think that nicely wraps it up for this webinar. I again want to abundantly thank our speakers, Daniel and Luisa, for their sharing their exciting work with us in a very clear and insightful way.
Thank you very much.
Yeah, and thank you to everyone who joined for the webinar as well. Just one last disclaimer that we're still looking for speakers for the next seminar as well, or the next semester as well. We will still send emails out about this, but do feel free to contact me or Rafa already if you're interested in giving a talk, such as the one that we saw today, the ones that we saw today. And then for the rest, yeah, I wish you all a very nice summer as well.
So thank you. Hi, Conchi, if I may, I see her. Conchi Atson. Hi, Conchi. I profit. So thank you very much for these initiatives. So I hope I can join the next one. So enjoy holidays, hopefully.
Yeah.
So,
Lisa,
Thank you.
Lisa,
Yay!
nice to see you. I have
Yes.
enjoyed a lot your talk. Very, very interesting.
Thank
And
you.
it has
Thank
been
you.
very nice to see you.
Yes. So I know you're still in the lab.
Yeah, still I am
Working.
here.
Working very hard. Yes.
But today
He's
I will
still very
start
active.
anyway. Yeah.
Okay. So good.
Very
Good
nice
to know.
to see all of
Like Paula.
you.
So
Yeah.
so
Yeah,
thank you. So
give my
really.
best regard to all the colleagues in Turin.
Of
Okay?
course. Sure. Yes.
Thank you.
Definitely. Starting from Paula. So
Okay,
so
very nice to
thank
meet you,
you.
to see you.
Yes,
Bye-bye,
thank you.
bye-bye.
Bye-bye. I leave.
Bye.
Hi, thank you, Naomi and Rafa. Thank you.
Thanks to you, Danny.
Thank you guys so much.
Also, yes.
Great talks, actually. Thank you
Really
so much.
fascinating.
We really enjoyed it.